Welcome to the PhD in Educational Neuroscience Distinguished Lecture Series, hosted by Gallaudet University. Welcome to those here in the auditorium and also those live streaming from across the country. This lecture series aims to honor world-renowned scientists in the field of psychology, education, cognitive sciences, and the neurosciences. These different fields, all interdisciplinary fields and those in between, contribute to the new and growing field of educational neuroscience. They increase our understanding of the human mind and neural mechanisms of learning. This year's distinguished lecture theme is From Mirror Neurons to Society, How the Brain and Experience Provide New Insights into Learning. With our distinguished lectures, here in the heart of DC, we want to build bridges across communities, countries, and everyone is welcome to attend. We hope that you will enjoy this distinguished lecture. Dr. Rizzolatti is our first fall lecturer. We are delighted and honored to have Dr. Rizzolatti accept our invitation. Professor Emeritus Dr. Giacomo Rizzolatti has received his degree in medicine from the University of Padua, Italy, where he specialized in neurology under the supervision of Giuseppe Moruzzi. He is currently affiliated with the Italian National Council for Research in Neuroscience in Parma and the Department of Neuroscience at the University of Parma. He's also members of numerous and prestigious scientific societies, such as member of the American Academy of Arts and Sciences, foreign member of the French Academy for Science, foreign member National Academy of Sciences. Dr. Rizzolatti has received numerous awards, such as the Brain Prize in 2014, which is one of the most prestigious awards one can have in neuroscience. He's also received numerous Italian national prizes and many international honors, such as the Prince de Asturias Prize for Science and Technology in Spain, the Prix Signore Neuropsychology Foundation, IPSEN, and the Gromeyer Prize for Psychology, University of Louisville, USA. Dr. Rizzolatti has been cited, at last count, more than 115,000 times, with at least 30 papers, garnering over 1,000 citations each, and growing. Dr. Rizzolatti is internationally recognized for his discovery of mirror neurons, a set of neurons that fire both when individuals produce and observe similar actions. So the exact same neurons are stimulated when one sees somebody picking up an object as when they pick up an object themselves. This discovery sparked research in the relationship between cognition and motor systems, and he subsequently went on to show that mirroring is a basic mechanism with a wide range of human social interactions. Today, we will learn more about Dr. Rizzolatti's mirror mechanisms, the motor system, and its role in understanding action and emotion. The title of his talk is The Mirror Brain. Now, please join me in welcoming Dr. Rizzolatti. And thank you so much for joining us. We are thrilled to have you.
which maybe will be superfluous for some of us, for some of you. But I think I have to say a few things. So the neurons speak one to another, talk one another by generating electrical impulses. They are called action potentials. You see here some of them recorded from a peripheral nerve. If I put an electrode here in my visual cortex, I will find the same potential, but they will be responsive to visual stimuli. If I put an electrode in my motor cortex, will be again action potential responding to movements. And the code is simply frequency modulation. So they will increase their frequency or decrease their frequency. That's the basis. So we use this technique, which has been developed in last century, for recording single neurons from the motor cortex. Here is a slide which show you the motor cortex, which is on the left side, where it's written F1, F2, and so on. And well, at this time there was a lot of discussion on how it was organized, so we decided to study this cortex, but we used an approach which was different from the traditional one. Instead of studying acceleration, velocity, trajectory, in other words, the classical aspect of the motor system, we used an ethological approach. We examined what the monkey does when we give food to her or break a thing or play with her. Anyway, it was just playing with the monkey instead of using the monkey as a small robot. Well, the results were really surprising. The first one, which you can see here, was the most neurons in the premotor cortex, at least in the ventral premotor cortex, do not fire in response to movement. By movement I imply that, a displacement of a joint. But they fire when the monkey organizes several movements in order to reach a goal. So in other words, this neuron here, for example, encodes grasping. Grasping done with the right hand, with the left hand, or the mouth. It doesn't matter. So you see here I introduce a concept which is different from classical motor concept. It's the goal. Here the goal is encoded. This neuron encodes the goal of grasping. The second surprise, I think due exactly because we used this ethological approach, was the discovery of neurons which fire when the monkey sees an object and is completely still. Imagine they have a neuron which fire when the monkey grasps in this way, precision grip. If you show a small object like a seed, there is a discharge. The monkey is doing nothing. So if I have a neuron which fires when the monkey grasps in this way, a big object will be effective. So in other words, there's transformation of object to movement, or to be more precise, from what is called the possibility of grasping an object, which are the possibility of grasping an object in the movement. We studied this phenomenon together with a Japanese scientist, Hideo Sakata, who studied the parietal lobe, and he found that in parietal lobe there is this neuron. So we traced the circuit which transformed what Gibson called the affordances of an object. For example, the affordances of this is this, or this. I cannot grasp like that. So the affordances of this object are transformed into action. That was the first discovery, and these neurons are called canonical neurons. The surprise for us and for everybody was that there were another category of neurons which were not interested in objects, but in the action. If you look here in B, there is a discharge due to monkey grasping. These are those action potentials that I showed you at the beginning. But if you look up, it's a person, an experimenter who is grasping, and yet there is discharge. So the same neuron fire both when the monkey is doing something or observing something. And the action should be a congruence in the goal. these neurons now are known as mirror neurons, and I have a film, but unfortunately it doesn't work, the sound. I will show you the film without sound, but it's a bit stupid. I don't know why the sound doesn't work. But you can see here, if you have a sound, here will be monkey grasping, will be a kind of
Fanno un altro così?
That's a grasping, and every time it's grasping will be
Come prima.
spike action potentials. And here is experimental grasping, and it will be exactly the same. If we could hear it, it will be the same.
Fai ancora?
It's a kind of dialogue. It doesn't matter who is grasping. And now look. He's putting his mouth. And exactly the same, because the goal is the same, it's grasping. So it doesn't matter if it's grasping with the hand or the mouth. And it's highly repeatable. We don't need analysis of variance or difficult statistical tools. It's there every time that you see there is a discharge. Well, there are two categories of them. One, which we call it broadly tuned, which is much more common in monkey, and the other are strictly tuned. If you look at these slides, you see that in terms of action, it's very specific. Precision grip determines this big discharge. Whole hand, nothing. But in terms of observation, it's the same, it's grasping. So that's we call it broadly tuned, because from the motor point of view, it's extremely precise, but from the point of view of vision, it's enough that it's grasping. In humans, I think they are much more strictly tuned neurons, but not in the monkey. Another important point is that this has nothing to do with conditioning. In this experiment, we introduced a monkey who has nothing to do, is an intruder. You see in A, this monkey sitting there and eating. We record from another monkey, which in this case is a grasping neuron. Here the monkey is grasping and good discharge. The experimenter is grasping is good discharge. And also when the intruder is grasping, but notice that the monkey from which we record is not rewarded. So reward has nothing to do with this. Simply, every time there is this type of action, there is a discharge in the brain of the monkey. All these experiments are rather old, and has been done with single neuron. So you put an electrode in the brain and record a single neuron. Now, fortunately, technology improved very much, and we have this type of electrode, like this one, in which we can record from the shaft. And so we can record simultaneously many neurons. And so we have also some number. That has been recently published by myself and a colleague. I don't say the name to not make difficulty for the translator. And you see here that it's a considerable number of neurons. The majority are, of course, purely motor, but a considerable number of neurons are either canonical neuron or mirror neurons. Now, the big question is why we have this neuron, why the visual system is not enough. Here is a sentence written by Marc Généraux. Marc Généraux, I think, Marc Généraux died a couple of years ago, and I think he was really the precursor of all this idea that motor system is important for cognition. We were good friends, and I like to quote him because he was also a very good writer. And he wrote in 204, the perception of an action without the involvement of the motor system would only provide a description of the visible aspect of the movement of the agent, but it would not give precise information about the intrinsic component of the observed action, which are critical for understanding what the action is about, what is its goal, and how to reproduce it. So here it's very important, this last sentence, how to reproduce imitation. Today I will not talk to you about imitation, but mirror neurons are very important for imitation. And the other point is to understand really what the action is about. I think the best experiment which explains that is an experiment made in Holland on children. Harold Beckering did an experiment on kids when they were still unable to walk. So a kid who is unable to walk, if you show another kid walking, there is no mirror neuron response at all. But if you show to the same kid, another kid crawling, there is discharge. As soon as it grows, as it learns how to walk, there is discharge also when it is observed another person walking. So that's this important message, that the importance of learning something allow you to understand much better what it is. And that is true also for adults. Just to quote, I am jumping ahead, I will show you in a moment the evidence that mirror neurons exist also in humans. But another experiment which I like to quote is done in London about the people who dance. The classical ballet people, if observe another dancer, have a strong activation of mirror neurons, which is almost lacking if you are not expert in dancing. If you show somebody dancing capoeira, the Brazilian dance, the war dance of Brazilian, there is very little activation. So the activation, if because you know to do something, and there is many experiments of this kind with athletes, with piano players, and so on. So learning something allows you to understand in a precise way what is going on. It's not just that you know. Sometimes rules are involved with which play a role, but even without rule, it's very clear that if you learn something, you understand it much better, rather if you don't know how to do it. Well, up to now, I have spoken about single neurons. But if we want to have a circuit, we want to have to use brain imaging. And we used brain imaging not many years ago. First we used it in humans, you will see in a moment. But we use it also in the monkey. In the monkey, it's very difficult to use brain imaging because probably everybody knows. If somebody of you have done this experiment or be a subject of that, the first thing the doctor told you, don't move. Because if you are inside the scanner and you move, the magnetic field change and it's full of artifacts. So you cannot do it. So how to do with the monkey? So it was a very good idea in Belgium by Guy Orban. So they put the monkey in a box, I don't remember how it was made. Anyway, in something was not a fake FMRI. So the monkey was sitting there, fixating a point, and when the point changed the light, it was reinforced. So the completely still and learned to stay in this position. Well, after 3-4 weeks, the monkey liked it very much to go inside this box in order to be rewarded. When this was done, at 5:30, 6 o'clock in the evening, when the patient are out, you bring the monkey inside the neuroradiology, and you have a cheap way to, I say the cheap way because in Germany they spend a lot of money in order to make rather complicated stuff in order to study fMRI in the monkey. However, that was a cheap way and it works very well. So we did this experiment in collaboration with the group, the Belgian group. Here it's the title. Here you see also the name. Guy Orban was the leader of the group, and Ken Denison was the student which really had deserved a lot of merit in this technique. And I don't present you the original data because it's no point here, but you see the circuit of mirror neurons in the monkey, which start from temporal lobe, that's the superior temporal sulcus, the area studied by David Perrett, then it goes to the parietal lobe and go to the motor. Here we don't have mirror neurons. It's a purely visual area. The mirror neurons appears in the parietal lobe and they became rather frequent in the premotor cortex. Now, of course, let's move to human. To human, we were very lucky because in '96, when we discovered this neuron in Milano, they just organized a very good PET center. And I asked to the director, Fazio, if I can do experiment there. He said, yes, sure. So we did experiment in Milano, and we found what you see here. So that also in humans, if you put the student inside the scanner, and in front of him you do movement, you break things, and so on, you have an activation of the motor cortex, which was a bit surprising, because it's a motor cortex after all. This experiment has been repeated several times. By myself, I went to Los Angeles working with Scott Grafton and Larby, but I want to show you something which has been done in Jülich in Germany by Karl Zilles and his group. It's the best anatomy that we have in the world, I think, in Germany. Well, that has been done in Jülich. It's a meta-analysis on about 90 patients, I think. And you can see the activation. It's very similar to that I showed you a moment ago in the monkey. STS, parietal lobe, premotor cortex. It's very big, very large, but essentially in evolution, the same area which were active in the monkey are active also in humans. Now, just a small parenthesis. Why there are so many areas interested in action? What's the reason? Now, a story. Somebody of you remember Nick Humphrey and his work on the visual system. Nick Humphrey, at a certain point, has a kind of sentimental problems. He left England, he was professor in Cambridge, and went in Africa, went to Rwanda to study gorilla. He studied gorilla very well, very carefully, he was a good scientist. Then he wrote a book, and that's from the book. There was no obvious sign of the gorillas using their intelligence to any practical advantage. Hard as they looked, I never saw them do anything that struck me as being clever, let alone any sign that they were having to solve difficult conceptual problems. So here there is an interesting problem. Why gorilla have a big brain? According to Darwin, every organ which is large, active, should have a function. Why gorilla have a big brain if they don't do anything? And he's right. What he described is gorilla wake up in the morning, they have big animals, they have no enemies, plenty of food, they walk around, eat, have sex, go to sleep again, and then the next day starts. So why the brain? Why to pay a lot of money, money in terms of energy, food, and so on for your brain if it's almost useless? And here is interesting picture done by him. I wish I know why the brain. But actually, pathologists, the answer is clear. What is complicated in the life of gorilla? They have not to solve mathematical problem, they have no conceptual problem, they have no enemy, they have not to organize an army. So, but why they have a big brain? Because their social life is complicated. So they are subdivided in big parties, in small parties, and one is fighting with the other, and one group is against the other, and the boss have to maintain the position of boss. And I have been told, I think it's true, that when they kill a big animal, they give the meat first, the boss, give the meat first to to the smaller people, and then he eats last. So to somehow it's kind of bribe in order to remember that I am a good boss. So the brain of primate has been called Machiavellian brain by people in Edinburgh. So it's really a brain which is done not in order to solve mathematical problem or philosophical problem, but in order to have social life, somehow to survive, to be a boss, to have social life. Well, let's go back to science. After our discovery, there was a lot of discussion, but these mirror neurons, why are there? For example, the birds. The birds are able to fly, and still we don't. So why the motor system should be so important in order to understand action? So we did an experiment which is really important. At the beginning, we have not understood completely its importance. So, it's fMRI experiments, which I do with Buccino. And the experiment is subdivided in two parts. In the first part, fMRI experiment, students lying in the scanner, and he sees a movie in which a student is biting, or the monkey is biting, or the dog is biting. So they are doing something which is common to all three species. Second part of the experiment, they see something which is specific for one species. For example, the students see a boy which is reading, or the dog which is barking, or the monkey which is doing lip smacking. Lip smacking is an affiliative gesture the monkey does, to say, I am your friend, be quiet, and so on. So the results are completely different. What happens if you have biting? Something which all species have. If you look at the left hemisphere, which is here, look, increase the blood, it's exactly in the same point. If I do subtraction, the here will be 0. So when I see a dog biting, I map it on my capacity to bite. It's exactly as if a human being were biting, and the same for the monkey. But for the other type of action, it's completely different. You can recognize on the left Broca's area. Laura, I think you have no doubt about that. You have Broca's area active. But look here. When the dog is barking, you have activation of the visual area, but nothing else. So how we recognize? We make the briefing of these people. We ask us what you see and so on. They said, well, the dog was barking. Obviously it was barking. There was no noise. Barking. So how they know it? But that's just called inferential thinking. So in the same way as you will understand why the apple is falling down, or the bird is flying, or airplanes are flying. So we have two distinct ways to understand action of others. And that will be even more important when I will talk about emotion. One, it's related to myself. It's mapping. He is like myself. I am biting, he is biting, it's the same. The other is something which I don't know how to do it, but I understand because I can make a logical thinking, I make inference, and so I know that when the dog is barking, he is expressing something. Just a small anecdote, a French philosopher wrote me that he asked his son if he would like to be a dog, and the son said, no. Why no, he said, because I will be unable to move the tail. Look, it's a very deep thinking, so you cannot be a dog because you don't know how to move the tail. Okay. With Corrado Sinigalia, he is a philosopher in Milano, we call it to mapping on ourselves, understanding from the inside. Now I move on something which we did more recently with our emotions. And the area that we studied is the insula. Insula is a part of the brain which is hidden between parietal and temporal lobe. If you open parietal temporal lobe, you see the insula. With two students, we stimulated electrically the insula, because the insula, I think so many stupid things has been written about the insula. It's one of the areas in which, I don't know, nobody wrote stupid thing about visual cortex. But about insula, you can read everything. Is this the place where the soul is, the personality, memory, everything is there. So we did a very simple experiment. We stimulate the insula using prolonged stimulation, like Michael Graziano and Charlie Gross did in Princeton some years ago for the motor cortex. Well, now I will show the result of the part in which I am interested. So here there are many colors, but That's because they are different functions. But look in B. In B, you have two big areas. One is red and one is blue. I will show you what happens when we stimulate the red area. And what you have to do is to look at the light. At a certain point, the red light will appear. It means that we are stimulating. And look at the behavior of the monkey. It's very simple. Red light. Red light. Red light. Well, I think it's obvious. Every time we stimulate, the monkey starts to make ingestive gesture. He's moving the lips, moving the tongue, maybe also doing something inside the tongue. Anyway, that's obvious. By the way, people recorded from this area, their neurons, they are related to mouth and to texture of the food and so on. Anyway, ingestive, positive ingestion. Now I move down, and I will be now in the blue area, more ventrally. And now look again. Red light. She's not happy. Now we will give some food to her. That's a nice fruit. No. She doesn't like it. Surprise. This food, which is good, is now not good at all. Again, there is a kind of, on one side, I see something which I like, but it's something now, it's disgusting for me. You see the surprise that something which was from 1 second ago was excellent, and now she throw away. In other words, you have a dorsal part which is a positive, and the the ventral part which is a negative. There is again a meta-analysis made in Jülich which indicate the ventral part of the insula is related to negative emotion. So, but anyhow, remember, so the dorsal part and the ventral part. One is positive ingestion, the other is negative disgust. Now, let's move to human. We studied disgust in humans in Marseille with Bruno Vicaire and other people which I don't mention in order not to make confusion. And that was fMRI experiment. Why we went to Marseille? Because in Marseille there was a group of people which were great in odorant. Odorant is very difficult for physiologists to master because you don't know how much you do, it's volatile, go everywhere and so on.
Using what? I missed it.
It's volatile, it go around. Odorant. Odorant, something that you put in the nose. So it was this experiment. There was a guy just lying in the scanner with a cannula in the nose, and they put inside different type of odorants. They could be disgusting or pleasant or neutral. So I repeat the experiment. The student is in the scanner and he has some odorant in the nose. We are interested now only in one type of odorants, and those which produce disgust. And the best of all are rotten eggs. Rotten eggs is something which nobody said is pleasant. Everybody says it's disgusting. Okay, now let's see the result. This is the first part of the experiment. The second part of the experiment, the student, again lying in the scanner, see movies in which there is a person, an actor, this actor here that you see behind me, which expresses some emotion. Remember, this guy on the left is not a disgusting gas. He expressed disgust, but he's not disgusting. So, normal person, but he is expressing disgust in his face. Now the results. The first part, look at B. You see this red spot, that's the rostral part of the insula. As in the monkey, disgusting stimuli activate the rostral part of the insula. That was not original data. It has been found in England by Phillips and the other before us. Anyway, we also found that if you gave a disgusting stimulus like the rotten eggs, odorants, you have this red spot, clear. The interesting result was the following, that when the student saw a face expressing disgust, they have activation of the same voxels which are activated by natural disgusting stimuli. This white spot that you see here are the same voxels. You see what it means? It means that it's not that this guy understood that the person have disgust, but in his brain fired the same neurons which fire when he is disgusted. So it's the same experience. We are in the same state. So that's really the capacity to understand the condition of others in an empathic way, not by reasoning. We can do by reasoning, of course. I will do some example. But you can do also in this direct way. And the same is true for, with pain, there is a lot of confusion. It's another field which is rather confused. But anyway, with pain, there are data from neurosurgery, the same. You see here on the right, this action potential, we are receiving, it's written. It's the patient which receive a painful stimuli. I have an activation of a neuron in the cingulate. I have explained later what is cingulate. And on the watching the patient is watching the neurosurgeon punching himself. So also in this case, you have this capacity to replicate in some other cell the action of the others. But so remember, I told you that we have two ways to understand the action. One is by activating directly the same structure like here, which produce pain or produce disgust or produce fear or produce many other things, or in another way. When I said that, because there are data from patients, for example, from the group of Damasio, Adolphs, and the other, they are patients with lesion, bilateral lesion of the insula, which don't recognize disgust. In their terms, the patient see vomits. They ask, what do you think about that? And he said, delicious. So you see the capacity to distinguish what is pleasant and lost. But the point here, it's more important, have a lot of political implication. Because imagine, first of all, society, ideology, can modify what is biologically determinate. For example, the neurosurgeon or the surgeon in general, when do an operation, can see blood, can see something terrible, but he doesn't stop. He is not in pain for this reason. Or even a more strong example, think about special forces, the policemen which have been trained to beat the demonstrant. Well, they don't stop when they see somebody crying or some blood. We have the capacity to block, to prevent our natural capacity to feel like the other, empathic with the other people. But now, imagine for a moment that the same thing has been done not by like surgeon or policeman, by ideology. And I like to remember you what happened in Germany in the 30s. Germany is one of the most civilized countries in the world. And this country was very civilized also in the 30s. It's the country where was born Goethe, Beethoven, Hegel, many scientists. I don't think there are so many countries so civilized like Germany. And yet, something happened. How it's possible that in the 30s there was a persecution of Jewish and they were killed? So speaking with sociologists, the explanation is the following. Germany lost the war, and other countries were so stupid to make some sanzioni, come si dice, some punishment, so the Germany was in a very poor condition. At this point, there was a genius, I am sorry to use this term, it's Goebbels, which made a propaganda saying, how it's possible that our country, which is probably the most civilized in the world, lost the war. There is something wrong here, there's something not true. So the idea was there are traitors among us, and the traitors are the Jews. So they are traitors, and so they are practically, the term which has been used by Hitler was Untermenschen. They are not really, they are primates, they are not human beings. And if you read the book on Eichmann process, you will see that this stuff which I discussed with sociologists and they supported my view. If you read the book on Eichmann, Eichmann has been examined by 3 psychiatrists, and all of them said that he is a very nice guy. One said also that he is an individual with very noble sentiments. He likes the father, he likes the children, he likes the wife, he likes the country, he likes nature, he likes animals. So all the elements in which we think. So why he did what we think was very bad? Because he was considered there was nothing wrong in what he did. If he said, that's his word, if I have been ordered to bring cattle from Budapest to Auschwitz, I will do it, because that's my role is to organize transportation. They ordered me to transport Jewish from Budapest to Auschwitz, I did it. Because for him, there were Untermenschen. If you don't consider that the other guy is like you, this is almost obvious why you don't have to do it. It seems strange, but our basic mechanism should be reinforced. And then I will show you these sentences from Bible, in which the people already in those times were sure that if you don't reinforce this basic mechanism, you can do horrible things, because if the other guy is not like me, why don't kill it? I can kill you. I kill the animals, I cut the trees, I can do it. Okay, now the last part of my talk is something that we are doing recently, and something which I am very happy to do, and that's after 15 years, that everybody was extremely happy with FMRI, with PET, saying, oh, beautiful, we can read the brain, we put the patient inside the scanner, and we can read it. I remember when I read the first paper by Roland, in which he said, motor imaging produce activation in the motor system like an action. I was elated, wonderful. Well, what happens now that we know much more about fMRI? So here are 3 points which are crucial. It's a non-ethological condition. As I said before, the doctor said, be still, don't move. So how you can study motor system, how you can study laughing, how you can study many emotions if you have to stay still inside this machine. Second, the data are only correlative. If you find an area which is active because there is painful stimuli, you cannot claim this is an area for pain. Maybe, maybe not. You must have additional data. And the third point, which again, people somehow have completely forgotten, it's time. Time, we live in a 4 dimension, not in 3 dimension. So time is extremely important. And yet, all our data, all our fMRI data are just picture, are not movie, are picture. The time has been disappeared. Now I'll show you how we can go ahead of this. It's called stereo EEG. This is a system which has been developed first in France by Talairac and the surgeon there, and then in Italy. Now there are few centers, one is Freiburg in Germany, but most of them are in France and in Italy, in which they study people with epilepsy using stereo EEG. Well, typically, epileptic patient can be cured, or at least the seizure can be stopped using some drugs. But in 20-25%, they are resistant to drug. So you have a poor person who have epileptic seizure and is not responsive to drug. So the only way is to find where is the focus inside the brain, to eliminate it, and in this case, the patient is okay, it's fine. But how to localize it? The old system was to put electrodes on the brain to see the EEG, but it's so approximative. So the stereo EEG is a system which provides the possibility to localize precisely where is the focus in the brain. And you see here, the first thing is they have to put many electrodes in the brain. This red spot are electrodes of the type which is shown there, but I already showed you before that we use in the monkey. So usually in Milano they put 16 electrodes in the brain. Each of them have 10 leads. So you have 160 points from which you can record from the brain. Then you do structural MRI in order to be sure there is no deformation of the brain and so on. And that's angiography. Angiography, it's, you put some colorant inside the vein, so you see how it's organized. And then very carefully they insert electrode. Here it's a picture. That's angiography, and that's the electrode inserted in that. Well, you do that, and then when you put this electrode in the brain, you wait for 4 or 5 days. During these 4 or 5 days, you record continuously, day and night, EEG, and you have a video. So you are sure that you will not miss an epileptic fit, seizure. After 4 days, typically you know where is the focus. You put away this electrode, you close it, you wait for a few days, and then you eliminate the focus. And the success is enormous. It's 95% of people after the surgery, they are fine. They are normal. They have no epileptic. But what is the advantage for a physiologist? The reason why we have been contacted by surgeons is saying we have plenty of data on normal brain which have no time to examine, to study, because we are busy to do neurosurgery. But many of these electrodes are in part of the brain which is completely normal. So one can study them, and I will show you some example of that. One is the following. There is one additional thing. Of course, nothing is easy in science. If you use this brain, which we like very much, most of the gray matter is inside the sulci. So we use the flat maps. So the brain is opened, and you can see also the gray matter, and that's also facilitate to put one brain on the other in order to have many brains. So now, the first experiment I show you, it's a median nerve stimulation. That's something that surgeons do for clinical reason, to know where is the localization of the median nerve, where is the cortex localization. So to do an experiment, however, we have to, let me do that. You see, you must have many patients. That is an experiment we started 5 years ago, and now we have 96 patients, and we have 11,000 cortical leads. So really, you have a very nice map of the brain for a very simple thing, stimulation of a median nerve. Now, look what happens. If you stimulate median nerves without time, there is no time. Stimulation of the nerve, you don't care about time. Well, you see activation, this yellow spot in the primary somatosensory cortex. But the picture is completely different if now I introduce time. If I introduce time, after 20 milliseconds, I have the classical picture that we have in the neurophysiology book. So activation of the area 3A, 3B, 1 and 2, and so on. But after 40 milliseconds, the stronger activation, the yellow spot, is the premotor cortex. But look here, after 100 milliseconds, we have a strong activation in the periciliar region. So that's certainly not for knowing that you have been touched. It's something different. According to some neuropsychologists, in order to be aware of your arm and so on. You have continually which render you aware of that. But it doesn't matter the explanation. But look the difference. If you consider fMRI, which is, give the same picture I show you just a second ago, and if you look at the time, you have area which you don't imagine that are activated. Now time is running, so I will show you another thing which really allow you this method, intracranial stimulation. That's something that we published just a few months ago on brain. We studied the whole cingulate, again, thanks to surgery which provided us with a lot of data. The cingulate is this gyrus which is in color. There are many different functions in this case, but look at the blue area which is area 24, the caudal part, AMCCV, and the other. Now I will show you what happens when here we stimulate. It's exactly the same experiment like the monkeys. This time it's a human being which is stimulated.
1, 2, 3, 4, 5, 6.
Che c'è? Cosa è successo?
Tu volevi andare via?
Cioè?
Well, unfortunately there is no sound, but what he's saying, I want to go away, I want to go away. And the same thing has been described by Talairak, an urge to move. So the stimulation of this area is an area which somehow gives you a stressful condition. You want to go away. By the way, that's interesting because many people consider this an area for pain. They say, oh well, because it's true, if you give a painful stimulus, you have activation of this area, but not because it's painful area. The same surgeon, I show you the data before, stimulate this area, never found pain. So that's correlation. So correlation is wrong. Correlation says pain, but if you stimulate, there is no pain. It's an area in which you want to go away. It's an area for going away. It's a motor area, very elementary to go. Now, that's the last thing which I show you. We stimulate also the rostral part, which is indicated as PACC. Again, you will not hear what's going on, but you will see. Here is a girl, which we stimulate this part of the brain. She's saying this thing. What she's doing? She's laughing. So every time when we stimulate this region, in many patients, we have now several of them, the patients start laughing. And if you ask why, there is two categories. One say, because something funny came to my mind. The other say, I don't know. I was compelled to laugh. Before commenting, I will show you something. We, with Fausto Caruana, we decided to see what happens if you show to the same patient whole stimulation determine laughing, these faces. One is sad, one is laughing, that's moving, one is laughing, and one is well, look at the red line. The red line is stereo EEG recording of high frequency gamma, and every time when you see laughing, you have an activity exactly of the same region whose stimulation determines laughing. So you understand that somebody is laughing because it's exactly your brain is laughing in the moment. You have the same phenomenon they show you for disgust. So we understand laughing because we are laughing. Of course, that's not the only center for laughing, because from there goes down a lot of pathways going down. But this is the director of the orchestra. The director is there, and one is the activity of this center start. It's coming back to the other stuff here. I think I have to stress again that many people use fMRI, to be very careful to not believe that if you have an activation of an area, therefore this area for something. If you read the literature of FMRI from 2000, 2005, you find area for everything, for all higher order cognitive function, which is not true, absolutely not. They are activated, God knows why. For pain, I show you because we know. But other, for example, a person which I admire a lot because he is a great psychologist, Michael Posner, said that that's important also for Stroop effect. Because he did Stroop effect, this area was active, but that's not this area for Stroop effect. This area completely is for when you are stressed. And if you have to resolve Stroop effect, you are stressed. You have an activity of this area. So, to conclude, first, we have new methods to study, and we have to use it. Third, this method confirms our idea about the mirror stuff, because laughing and pain, not pain, but anyway, conditions similar to pain, are determined activation here. And with laughing, it's extremely strong. Okay, thank you.
Thank you.
Thank you for sharing your amazing work. It was a pleasure having you here today. Sorry, I will start again. Thank you for coming here. It was a pleasure having you today. We've really enjoyed you sharing your work. Before we go on with a few questions, I have two small communications. As always, I will ask everyone here in the room to fill out our form on outreach. I will pass the papers around. And at the end of the questions, please join us to the reception in the building, the Science and Communication and the language building just on the other side. So do not run away. Please join us for more questions. So for questions, if you have a question, please come here so that it can be live streamed and everybody can participate.
You're going to translate it. Yes.
Hello, and thank you so much. So my question is, I just actually want to know your thoughts.
I cannot, I don't understand what she's...
She would like to know your thoughts on
So there are a few neuroimaging studies that have shown that deaf native signers, while watching sign language, don't activate their mirror neuron system.
What you will do?
And the hypothesis is that the more manual or the larger the movement is, there's a more direct path. So there's a more direct stimuli of the action observation. So there's a more direct path to action observation, the action observation system, more generally, without utilizing the mirror neuron system. So again, that's just a hypothesis. So I would love your thoughts on that.
Well, if I understood correctly what you're asking, you said that in the sign language they don't use mirror neuron. I don't understand the logic of that because, as a matter of fact, I think the language in general, I have not spoken about that, but our hypothesis about language in general, that it derived from movements. And the point that Michael Arbit and myself put is the following. If you have an action like that, you will understand, even if you are here or you are in another country, you are in Africa and so on, because what the sender said, the message, is present in the receiver of the message. So the sender and the receiver share the same thing. So that's the basis. Because otherwise, how it's possible that that's called battle? For which reason that's called battle? So it should be a basis on which the sound grows. So the idea is not original because it started with Condillac in the 18th century, and then Armstrong, Stokes, and many other people. But what we provided, some neurophysiological evidence that indeed we have a system which give to the sender and to receiver of the senses a common ground. So I think the sign language derived from that as our language. I mean, the root is the same. I don't understand why the sign language then followed another route. I have to add that also emotion played a role, that we somehow minimize it, but emotion also play a role in that. But it's fundamental. It's the other thing, the other reason why with Michael we proposed again this idea is that anatomically the pathways which put together Wernicke and Broca's, so the basic neural pathway, it's on the surface of the brain, it's not in the depth of the brain. All the animal calls and so on are due to stimulation of deep brain structure, which are related to emotion. Third, the human language, it's a dialogue. It's not related to emotion. And a point which was not raised by other, it's imagine a word like fire. Fire can have only one meaning for an animal, escape. For a person could be fire, give me fire for my cigarette, give fire because I want to do a barbecue, fire because there is really fire escape. So it's somehow this disjunction is not related anymore to emotion. So I think the sign language is right. Michael Arby, who worked on this point after me for several years, thinks that actually was a kind of proto-sign language at the beginning. The sign developed, it became very sophisticated, like language, true language became sophisticated. But the beginning, or the root of both language, sign language is the same. It's a proto-language that's related to our movement of the arm and so on.
Thank you.
That means thank you.
I think most of those studies involve fMRI, and so looking at the deep structures. Whereas we're in our lab, we're using EEG, and we found results similar to what you've been talking about today. So I was just curious about the fact that those were seemingly contradictory results and your perspective on that.
What to say?
So in our lab, we use EEG.
Yeah.
And with the EEG findings, we found things similar to what you just described here today, related to sign language perception,
well,
which I think is really neat.
Thank you very much. That's correct. I think, however, the first who described that was Ramachandran in San Diego, who showed that the mu rhythm somehow reflect the mirror neurons. So if you find the same thing in language, it's the same thing. Yeah. In sign language, it's the same thing. Okay, thank you.
Thank you so much.
But that microphone is not working at all.
Good evening. I'm sorry I'm not able to sign all my question. My ASL is not enough. I'm a PhD student from Portugal. I'm visiting here Gallaudet, and I worked as an assistant to a professor in Portugal that worked in neurosciences, and I'm very interested about that sentence that you said that children don't have the same discharges of mirror neurons when they can't walk. Unless they start to crawl, then they will have those signs.
Right.
So they show those signs. So when you talk about disgust, pleasure, and even the laughter examples, I was wondering what you would say about empathy. Is empathy a cultural construct, or is it an in our nature, in our brain. So, or are we acculturated
No, that's
to
true.
empathy?
It's extremely interesting question because there is a dichotomy, according to me, between action that we learn and something which we are born with. We don't learn vomiting. We don't learn crying. It's something that we have already inside. So I think for emotion, for empathy in this sense, we are already ready when we are born to understand the mother. And in this sense, there is a wonderful book by Daniel Stern who describes the relation between mother and child before language. So there is a capacity to communicate which is incredible. And that is related on this inborn mirror neuron. Then I think we have the capacity to learn new action. I mentioned dancing, but sport and so on. So more you learn, the more you understand what the other people are doing. So motor activity is very important in order to have more capacity to understand the other people. But in this case, you're right, I don't think it's inborn, it's learned. So we learn many things. And well, we are not inborn with the capacity to dance, especially classical dance, which is so stereotyped and so on. So, but that's an important point. We discussed it a lot with Corrado. We are now writing something theoretical, but it seems there are really a dichotomy between emotion, which we are born with, and action, cold action, as they are called. But you really, it's a very important point you touched.
Thank you.
So I have a question and then a comment as well. Meltzoff's work looked at infants' ability to imitate, and I think this goes to your question as well.
Yeah.
So now let me ask you, you talked about action perception and empathy. And all of these things being through a visual medium. I'm not familiar so much with emotions and how they activate in the brain, but thinking about the fact that these are activated visually, do blind people experience emotion less than sighted people? Also, going back to your hypothesis, the mirror neuron system underlies empathy, but if you can't see the action, how does that tie into your hypothesis?
Good question, but there are experiments in this respect. It has been done experiment in blind people, and they recognize action through audition and through tactile. So blind people have the mirror system and the activation exactly in the same area. It will be done by this person you met in Luca Petrini, which make experiment in blind people. So blind people have this, and I suppose it's true the same also for emotions.
Who's the person that did the studies on blind people?
It's called Petrini, P-E-T-R-I-N-I, and he's working in Lucca, where you have been last week.
So he's the one who did the
Yeah. When he was younger, he did the research. Now he's director.
Got it, thank you.
Thank you.
Okay, I'm going to build on these two questions. It's a little bit of a curiosity, maybe a little provocative. So we know infants, when they're born in the first weeks, they have the protrusion imitation. If you stick the tongue out, the baby will stick his tongue out back. So they're able already at birth to imitate simple protrusion, right? Then you said that if the baby knows how to do a specific action, they will have the firing appears. So if the baby crawls and sees another baby crawling, it appears. At some point, baby starts producing sounds. Before they can actually produce words, they babble and they make sounds, and they're able to make the sounds M-A, ma, 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 ma. And a lot of parents spend their days in front of their babies saying, "Mama, mama, mama," hoping that the baby will actually give it back. But that doesn't happen that often. And why is there? So they definitely know how to do the sound. They definitely can see it. They know how to imitate because the setup, the mechanical setup is there. Why don't they give back the sound?
Well, the first thing is about the methods of experiment. I don't know if this protrusion of tongue is just action without emotional content. Many people think that that is related to emotional content. The capacity to imitate sound, it may be many things. For example, I don't know if the maturation of the system is sufficient to produce, to imitate. However, the birds are able to imitate using the mirror system. So that's a very nice experiment made at Duke, in which they show that the singing birds imitate the sound. That's a very good system in order to distinguish group of singing. I don't know, I have never thought why the children cannot say mama, mama, mama.
Any other questions?
Hi, Giacomo. Welcome to Washington, D.C.
You're an expert of audition. Maybe you can answer the question.
No, I'm not able to answer that question. I have another one, which I'm sure you've thought about for a long time. People are still sort of fighting about the motor theory of speech perception. And there's now, one group that says there's a strong theory and a weak version, so the strong version would be involvement of primary motor cortex in addition to the areas that you've demonstrated, like Pulvermüller's work and Kate Watkins and so on. Evidence is actually not bad. There's TMS experiments where you can then inhibit primary motor cortex and it affects speech perception. So what's your opinion on that? And why would we need primary motor cortex in addition to the...
Well, what that are the experiment by Pulvermüller, essentially, I think. So the one you hear, for example, he is kicking beside activation of language area. You have also an activation of the motor cortex and approximately the area where is represented the leg. If you heard another word in which you use the hand, you have an activation. Now there are two possible interpretations. One is the small one. This is necessary in order to understand really what's going on. The other, it's not that this is necessary, but this increase your vividness of the fact. So even without motor cortex, you will understand kicking. But one thing is kicking with the motor cortex, which in the sense you have also the motor image of what's going on. The other will be only abstract, what is going on. So I don't know which is correct, also because the localization, it's not particularly nice. It's in the motor cortex, but it's a bit, honestly, I don't know. But for phonology, I think Lieberman was correct. Right. I mean, for phonology, there is no doubt there is Broca's area activation, if you say something.
We're doing monkey experiments. For a few years ago, when you invited me to come to
Elche,
yeah, I was the first report of that, and now we're sort of a little farther along. It's imaging, functional MRI in monkeys, as you mentioned. It's not easy to do, but I will share. I can't share the full results yet because we're writing it up right now.
Well, the essence.
But the essence is that we see motor activation in an auditory motor task. Obviously, monkeys don't speak. It's hard to get them to articulate, so we developed a different paradigm where we have them press levers so they can produce sounds, and they produce sound sequences or something that they can learn quite well. And then we put them in MRI and we play the melody to them, and they activate their motor areas. So
it's very interesting.
There's a new paradigm now that we can hopefully expand and use to test some of these ideas.
Here is Professor Rauschecher.
So unfortunately, the time is up, but if you have more questions, please come at the reception and you can have probably some time to chat a little more. Thank you very much for coming. We have a little gift for you. Please, it was our pleasure. Here. It's everything, not only the balloons.
Everything,
You get
not
the bag
only
also,
balloon.
yes. The bag is part of our little gift.
Well,
thank you gift.
thank
it was
you.
a pleasure having you, and now we shall all go and have a little refreshment all together. Thank you very much for
Thank
everyone,
you.
and thank you for those online.